I'm Mrs. Murdoch, and today we are going to do part 3 of the diffusion osmosis lab in the AP Bio lab bench labs. So what we're going to do is we are looking to find how much sucrose there is dissolved in the tissues of this potato. The mystery concentration of sucrose in potato cells. That is what we are trying to find. In order to do that, we got to get inside the potato. So we're going to get inside the potato. This is the fun part. So I'm going to cut some sort of thick slices of potato that kind of look like that. And you can see there's water in there. And if I really went in and got some tissue out and looked on the microscope, you would see that there are actually potato cells in there. And just like any cell, there is going to be some dissolved things in there. And we are particularly paying attention to the amount of dissolved sucrose. Plants use sucrose as a way of storing sugar. So there's going to be sucrose in here. So what I'm going to do to make some potato pieces for myself is I'm going to use this coring device. And this is kind of fun too. You kind of just go in and you make these little bits of they look like little stovetop cylinders. See that? With some surface area there. And I'm just going to make a few of these to give you an idea of how I set this lab up. So there's 1, 2, 3, 4. I'll just make 4 of them because that's what I put into each one of the cups when I started this lab. Okay, so I've got these 4 slices of potatoes and I'm seeking to find out how much sucrose is in them. So in order to do that, what I'm going to do is I'm going to expose them to some different solutions that have sucrose in them. This solution, now the colors are just food coloring, but I added different amounts of sugar. Okay, it's just table sugar, ordinary sugar that you buy at the store, which is sucrose, which is a disaccharide, and we learned about that. So this is the most concentrated sugar solution, sucrose. This is 1.0 molar. And then over here at the other end, this is just plain water with no sucrose in it at all, right? Okay, so now let's go over here and kind of look at the diagram of what I'm doing here. So all of these potato pieces have the same amount of sucrose in them. They're from the same potatoes. They're going to have a certain amount of sucrose concentration inside them. So here's my 6 cups that I've got. Here's my potato slices, and they've got some some sucrose in them, about the same. I'm going to try to make dots that indicate the same equal molarity, mystery molarity of sucrose inside these potatoes. And it's all the same. We just don't know what it is, okay? And then outside in this 1 molar solution where I put this stuff around the potato, right? So this is around the potato. I have a known amount of sucrose out here. So this is going to be very concentrated. 1 molar sucrose is actually rather syrupy, so there's a lot of sucrose particles in there. Whereas maybe in the 0.8 molar, maybe slightly less, but still pretty high. Okay, so the 0.8 there. And then as we go less and less, there's less sucrose molecules outside the potato slice. Here at all, and then finally that water is just tap water. There's nothing. There's no sucrose in there at all. Now remember, we talked about this before. Every time you have a cell in a solution or a potato in a solution, right? So if I do this, I've got 2 solutions to be concerned about in this cup. I have the solution that's red that's surrounding the potatoes that they're immersed in, that has a known sugar solution in it. And then I have the solution that's inside the potatoes, that's on the other side of a barrier called the cell membrane, and the cell walls are also in there, right? But the cell membrane is the one we're worried about because that's the one that's selectively permeable. That's going to let the water through it. The question is the water going to go into those potatoes or out of them based on the relative amount of sugar that you've got here? Okay, so you may remember words like hypertonic and hypotonic and isotonic are going to be at play here. We're just not sure which ones are which because we don't really know. This is still a mystery how much sucrose is in the potato. So now what I did was yesterday I did 2 trials of this. So I made a whole row, I made a whole row of cups of the solutions with samples of potatoes in them. And they've been immersed and soaking in there all night, and now it's 24 hours later, and we're gonna film me taking the final masses. Now this is what the potatoes look like in the solution right now when I just put them in. Look what the potatoes look like at the end of soaking in the really concentrated solution. You see the difference there? This looks a little bit shrunken, doesn't it? Doesn't that look shrunken to you? I wonder what that means. Okay, so here we go. So what we're gonna do now is I'm going to take masses, final masses, because I took the initial masses of all the potatoes, and now I'm gonna take the final mass. I'm just gonna do one trial, just so that you're not here all day. So final mass for trial number 1, and this is the ones that were soaking in just the ordinary tap water. Hey, look at these. Look how big and fat they are. Very different from the ones that looked all shriveled up that were in the concentrated one, aren't they? Yes, okay, so maybe that will mean something too. All right, so I'm going to just dry them a little bit, and I'm gonna try to do this the same for everyone. Tear this. Okay, here's my first mass. That's not tearing. Tear. There we go. Okay. And here's my final mass of those 4 together. It started at 8.9 and now is that 11.2? I think it is.
Yes.
Thank you, Mr. Bouchard. Okay, so get that out of the way. That one's done. Here's the next one. Block them just a little bit. Try to do them all the same way. That looks like 9.9.
Yes.
Okay.
We're switched to 10.
Yeah. Looks good. Okay, here's the 0.4 molar sucrose. They gained a little bit of that blue color. Which, as far as the years I've been doing this lab, the color doesn't seem to make any difference, so we're good with that. So that's his 10.2. Oh, did it?
Yeah.
Thank you, Mr. Bouchard. I'll make a 10.3 there.
Sorry.
No, please help me out because I'm trying to read upside down, so anything you can do to help me, that's great. Okay, here is the 0.6 molar solution soak. Again, getting slightly smaller every time. A little bit smaller there. There we go. 8.4, would you say? 8.4. Okay. What's that? What's fun about this? I mean, there's nothing but sugar water and food color dye, so don't be worried about me getting my hands. What my hands will feel sticky after this, though. Definitely going to have sticky fingers because all of this sucrose, we always wind up having sticky surfaces for a while after this lab when all of you are in here doing it. Once again, look at that. It looks a little bit more shriveled, doesn't it, than the other ones. They're getting more and more shrinky. Okay, 7. All right, I think we're on the last one. Here's those really shrunken ones there. They've been sitting in very syrupy stuff all night.
This is why there's ants in this room.
Yes. Yes, the little insects. Ants love sugar, and yes, when I have had ants in here, it's because of this lab. Even though I clean and scrub and clean with Lysol, even before the pandemic, I was being very clean in here, but it just, ants love the sugar. They seem to be able to smell it, whether I clean it or not. Okay, that's it. Here's the data. All right, now I'm going to pick this data point up so you can see. Just so you guys can see exactly what the data is. All right, there, that's trial number 1. Now I am going to be uploading this data for you into a Google Sheet, so I'll also measure those last trials. I'm not going to do it now. But one of the other data points that you're going to need to calculate, first of all, you're going to need to graph this data, and we're also going to add some other students' data from years past, so you don't just have a sample size of 1, even of 2 is kind of low. So we're going to make a nice big sample size for you, and then you're going to be able to figure out the sucrose concentration of the potato from that, and we'll talk about that another time. But in addition to that, you're going to use that sucrose concentration to help you calculate something called water potential. Water potential is the true goal of this. Plants tend to have this thing called water potential that it's a nice way of describing how much turgor pressure their cells have. And having turgor pressure in a plant is very healthy for a plant. So to calculate water potential, this is a calculatable thing. You need room temperature. So I'm just going to mention that right now it is about 23 degrees Celsius in the room. So in addition to that data, now you have the room temperature to deal with, which is part of the water potential formula, okay, which I will discuss in a different video. All right, and that is the potato soak data explanation and the data that you would need to discover the sucrose concentration of this potato and then calculate its water potential as well. Thank you, I hope that was helpful.